Hello students. In today's module, we'll be learning about the molecular analysis of photosystem 1 and 2. The energy conversions catalyzed by photosynthetic organisms are functionally divided into light reactions, which convert the light energy into an electrochemical gradient that is used to synthesize the high-energy substrates ATP and NADPH, and the dark reactions that consume ATP and NADPH for the production of carbohydrates by carbon dioxide fixation. The light reactions take place in the photosynthetic membrane and in large membrane protein complexes. During the last century, the sophisticated techniques of spectroscopy, molecular genetics, and biochemistry were used to reveal the structure and function of the two photosystems. The new structures of photosystem 1 and photosystem 2 from cyanobacteria, algae, and plants has shed light not only on the architecture and mechanism of action of these intricate membrane complexes, but also on the evolutionary forces that shaped oxygenic photosynthesis. Photosynthesis in the green and purple bacteria does not generate oxygen, whereas photosynthesis in cyanobacteria, algae, and higher plants does. This difference is attributable to the presence of two types of photosystems in the latter organisms. One splits water into molecular oxygen, and the other reduces NADP to NADPH. In contrast, photosynthetic bacteria have only one type of photosystem. Molecular analysis of photosystems. In the 1940s, biophysicist R. Emerson discovered that the rate of plant photosynthesis generated by light of wavelength 700 nanometer can be greatly enhanced by adding light of shorter wavelength. He found that a combination of light, like 600 and 700 nanometer, supports a greater rate of photosynthesis than the sum of the rates for the two separate wavelengths. We call this Emerson effect, and this has led researchers to conclude that photosynthesis in plants involves the interaction of two separate photosystems, referred to as PS1 and PS2. PS1 is driven by light of wavelength 700 nanometer or less, PS2 only by light of shorter wavelength, which is less than 680 nanometer. Photosystems comprise of reaction centers and light harvesting complexes, LHCs. Now let's look at photosynthetic reaction center. The photosynthetic reaction center is a multi-protein complex which is the site for the generation of high-energy electrons, a critical event in the light reaction of photosynthesis. The bacterial photosynthetic reaction center has been an important model to understand the structure and chemistry of the biological process of capturing light energy. The photosynthetic reaction center of purple bacteria has three integral proteins, L, M, and H. The reaction center protein contains a total of 11 transmembrane alpha helices, and to these the prostatic groups that absorb light and transport electrons during the photosynthesis are bound. The prostatic groups include a special pair of light-absorbing bacterial chlorophyll A molecules that are equivalent to chlorophyll A molecules in plants, as well as several other pigments and two quinones, QA and QB, that are structurally similar to the mitochondrial ubiquinone. Another component of photosystems Closely linked to a reaction center is an antenna complex, which consists of several protein complexes, including internal antenna proteins within the photosystem proper and external antenna complex called light harvesting complexes. Light harvesting complexes are made of specialized proteins which capture light energy and transfer it to the reaction center. They contain chlorophyll A and other photosynthetic pigments like chlorophyll B in vascular plants and carotenoids in both plants and photosynthetic bacteria. Now let's study the single photosystem of purple bacteria. The three-dimensional structure of photosynthetic reaction centers permits scientists to trace in detail the path of electrons during and after absorption of light. Now let's look at the initial charge separation. In the photosystem of purple bacteria, as in other photosystems, energy from absorbed light is used to strip an electron from a reaction center, bacteria chlorophyll A molecule, and transfer it via several different pigments to primary electron acceptor QB, which is located on the cytosolic membrane face. The chlorophyll thereby acquires a positive charge, and QB acquires a negative charge. Each pigment in the reaction center absorbs light of only certain wavelengths, and its absorption spectrum changes when it possesses an extra electron. Next is electron flow and coupled proton movement. After the primary electron acceptor, QB in the bacterial reaction center accepts one electron, forming QB-, it accepts a second electron from the same reaction center, chlorophyll, following its absorption of a second photon. The quinone then binds two protons from the cytosol, forming the reduced quinone QH2, which is then released from the reaction center. QH2 diffuses within the bacterial membrane to a Q0 site on the exoplasmic face of cytochrome BC1 electron transport complex, similar in structure to complex 3 of mitochondria. There, it releases its two protons into the periplasmic space, that is the space between the plasma membrane and the bacterial cell wall. This process moves protons from the cytosol to the outside of the cell, thereby generating a proton motive force across the plasma membrane. Simultaneously, QH2 releases its two electrons, which move through the cytochrome BC1 complex, just like that in the mitochondrial complex 3. The Q cycle in the bacterial reaction center, like the Q cycle in mitochondria, pumps additional protons from the cytosol to the periplasmic or the intermembrane space, thereby increasing the proton motive force. Electrons are then transferred from the cytochrome BC1 complex to a soluble cytochrome, a one-electron carrier in the periplasmic space, that is between the bacterial cell wall and plasma membrane. Now, this reduces the cytochrome from a ferric state to a ferrous state. The reduced cytochrome then diffuses to a reaction center, where it releases its electron to the positively charged chlorophyll A+, returning the chlorophyll to the uncharged ground state and the cytochrome to the ferric state. So here, the electron flow is cyclic, generates no oxygen, there is no reduction of NADP+ to NADPH, but it has generated a proton motive force. As in other systems, the proton motive force generated in the bacterial photosystem is used by the F0, F1 ATPase to synthesize ATP and also to transport molecules across the membrane. Now let's study the photosystems 1 and 2 in chloroplasts. As in the bacterial reaction center, at the center of each chloroplast photosystem is a pair of specialized reaction center chlorophyll A molecules, which are capable of undergoing light-driven electron transfer. The chlorophylls in the two reaction centers differ in their light absorption maxima because of differences in their protein environment. For this reason, the reaction center chlorophylls are often denoted P680 for PS2 and P700 for PS1. As in photosynthetic bacteria, each reaction center has an associated antenna that consists of a group of light harvesting complexes. The light harvesting complexes associated with PS2 and PS1 contain different proteins. The two photosystems are also distributed differently in the thylakoid membranes. PS1 is located primarily in non-stacked regions and PS2 primarily in stacked regions. The cytochrome Bf complex, which aids in the transport of electrons from PS2 to PS1, is found in both stacked and non-stacked regions. The stacking of the thylakoid membranes may be due to the binding properties of the proteins in PS2. And finally, and more importantly, the two chloroplast photosystems differ significantly in their functions. Only PS2 splits water and converts it to molecular oxygen, whereas only PS1 transfers electrons to the final electron acceptor, which is NADP+, and reduces it to NADPH. Photosynthesis in chloroplasts can follow a linear or cyclic pathway. The linear pathway can support carbon fixation as well as ATP synthesis. In contrast, the cyclic pathway supports only ATP synthesis and generates no reduced NADPH for use in carbon fixation. Photosynthetic algae and cyanobacteria contain two photosystems, which are analogous to those in chloroplasts. Now let's study the linear pathway of electron flow in PS1 and PS2. The linear flow of electrons involves PS2 and PS1 in an obligate series in which electrons are transferred from water to NADP+. The absorption of a photon by PS2 causes an electron to move from P680 to an acceptor plastoquinone, QB, on the stromal surface. The resultant positive charge on P680 strips electrons from the highly unwilling donor, water, forming molecular oxygen and protons. Now, these protons remain in the thylakoid lumen and contribute to the proton motive force. After P680 absorbs a second photon, QB- accepts a second electron and picks up two protons from the stromal space, generating the reduced quinone QH2. The reduced QH2 dissociates from its binding site on the PS2 reaction center and is replaced with an oxidized Q. Now, QH2 diffuses randomly in the thylakoid membrane, binding to Q0 site of the cytochrome Bf complex, where it releases its two electrons. The cytochrome Bf complex is similar in structure and function to the cytochrome Bc1 complex in photosynthetic bacteria and to the complex 3 in mitochondria. As in these systems, the Q cycle operates, thereby increasing the proton motive force generated by electron transport. After the cytochrome Bf complex accepts electrons from QH2, it transfers them one at a time to the soluble Q-prick form of the plastocyanin, reducing it to the Q+ form. Reduced plastocyanin then diffuses to the thylakoid lumen, carrying the electron to PS1. As in PS2, the absorption of a photon by P700 in PS1 leads to the removal of an electron. The resulting oxidized chlorophyll P700 plus, is reduced by an electron passed from PS2 via soluble plastocyanin. The electron given up at the luminal surface of P700 moves to the stromal surface of the thylakoid membrane, when it is accepted by ferredoxin, an iron-sulfur protein. Ferredoxin transfers the electron to FAD and finally to NADP+. Two electrons, together with one proton removed from the stromal space, convert each NADP+ and then to NADPH by picking up another proton from the stroma. P700 is restored to its ground state by addition of an electron carried from PS2 by plastocyanin. As in mitochondria, the movement of protons down their concentration gradient through an F0-F1 complex is coupled to the synthesis of 1 ATP from ADP and inorganic phosphate. Thus, this pathway exploits the energy from multiple photons absorbed by both PS2 and PS1 and their antennae to generate both NADPH and ATP in the stroma of chloroplasts, where they are utilized for carbon dioxide fixation. Photosystems comprise of reaction centers and associated light harvesting complexes. A reaction center is where the primary events of photosynthesis, that is, generation of high-energy electrons, occur. And light harvesting complexes are antenna complexes made up of specialized proteins which capture light energy and transmit it to the reaction center. Both reaction centers and light harvesting complexes contain tightly bound light-absorbing pigments. A single photosystem exists in purple bacteria that generates a proton motive force which is used for ATP generation, but there is no generation of molecular oxygen. Plants and cyanobacteria contain two photosystems, PS1 and PS2. They have different functions and are physically separated in the thylakoid membrane. PS2, which is similar in structure and function to the bacterial photosystem, splits water into molecular oxygen. PS1 reduces NADP+ to NADPH, which is then used for carbon dioxide fixation, that is, conversion of carbon dioxide to carbohydrate. And lastly, structural information of the photosystems is invaluable for our understanding of photosynthesis, probably the most important process on Earth. The information will also help design artificial photosynthetic systems for the improvement of bioenergy production and the enhancement of agricultural productivity. Thank you.